Neuropeptide Y promotes adipogenesis in chicken adipose cells in vitro, a comparative biochemistry and physiological study conducted by corresponding author Elizabeth Ruth Gilbert and fellow professors. Neuropeptide Y is a 36 amino acid sequence commonly known as NPY, found in the central nervous system, which plays a role in many mechanisms ranging from pain perception to blood pressure regulation, but most notably for appetite. NPY is produced in many regions of the brain, however, it is also able to cross into the autonomic nervous system. Adipogenesis is the creation of new adipose tissue which is filled mostly with adipocytes, a fat-storing differentiated cell. This tissue mainly functions as a reserve for lipids so that energy requirements can be met easily and effectively, even if there's a lack of exogenous glucose. Adipose tissue comes in two types, white versus brown, which store energy and generate body heat respectively. NPY has been extensively researched in the past, and many studies have contributed to the rationale that the NPY system in mammals can induce energy storage in adipose tissue and mitigate brown fat activation via both central and peripheral pathways. This study ultimately sought to explore the relationship between NPY systems and adipogenesis in avian species, based off the rationale that this system does correlate to adipogenesis in mammals. A fast-growing broiler chicken line was used as a model, and abdominal fat tissue was collected on day 14 post-birth to be examined. Prior to analysis of the tissue collected, researchers hypothesized that NPY injections would cause an accumulation of lipids in avian adipose tissue. In order to completely analyze the abdominal fat tissue, 7 sampling methods were employed, each building on one another, ultimately combining to depict the complete interaction and impact NPY treatments have on adipose tissue. In order to obtain a primary adipose cell culture, the abdominal fat tissue collected was put through the process shown on the slide. All experiments had at least 3 biological replicates with triplicate wells for each treatment of an individual chick sample for one experiment. The triplicate values obtained were averaged for each experiment, and the average was used for statistical analysis. Researchers employed NPY treatments of 0, 1, 10, or 100 nanomoles per liter, after which the cells were incubated in serum-depleted media overnight and finally harvested at 4 hours after treatment. Furthermore, the EDU complex allowed thymidine analogs to enter active DNA synthesis and subsequently Alexaflor 488 to detect it. NPY treatments were supplemented every day until the study was terminated. Oil red O staining, G3-PHD specific activity, and mRNA abundances of adipose-associated factors were used post-treatment to measure the extent of adipocyte differentiation. To obtain fluorescent images of adipose tissue, cells were isolated and upon confluence, induced to differentiate in the presence of NPY treatments. Cells were then stained with oil red O and counterstained with modified Mayer's hematoxylin. The red color captured indicates neutral lipids. In order to measure the glycerol 3-phosphate dehydrogenase specific activity in NPY-treated cells, lysates were obtained and then sonicated using the Bioreptor 300 and subsequently centrifuged. The supernatant obtained from this was used to measure the G3-PHD specific activity as well as the total protein concentration. The total RNA was examined using the RNeasy mini kit, the process of which is starred on this slide. The real-time PCR reactions were performed using the system shown, specifically with a melting step for 3 seconds and then an annealing/extension step for 30 seconds. Real-time PCR data was analyzed using the delta-delta CT method, while turkey tests were used to depict pairwise comparisons between treatment groups at differing time points. The ANOVA test was conducted using the relative quantity values obtained through the equation shown. Results. No effect of the NPY treatment was seen on cellular proliferation at 12 hours post-treatment, and similarly, there was no mRNA abundance difference for multiple proteins at 4 hours post-treatment. Conversely, with 100 nanomoles per liter NPY treatment, TOP2A and TP2X mRNA abundances decreased at 4 hours post-treatment. Simultaneous to this, NPY mRNA abundance was seen to increase. As the data displays, a significant increase in NPY2R mRNA abundance was seen at 4 hours post 10 nanomoles per liter NPY treatment. G3-PHD activity was seen to increase on day 6 post-differentiation, with the 100 nanomoles per liter MPY treatment activity level noticeably greater than that of the control treatment group. The images captured with oil red O display lipid accumulation found in NPY-treated cells at 2, 4, and 6 days after differentiation induction. As the images show, the lipid expression in cells appears to increase as post-differentiation time and NPY treatment concentrations increased. NPY treatments were seen to reduce CCAAT enhancer binding protein alpha on day 4, peroxisome proliferator activator receptor gamma on day 4 and 6, and steroregulatory element binding protein on day 4 as well. CCAAT enhancer binding protein beta abundance was seen to remain unaffected by NPY treatment throughout the entire 6 days. Fatty acid binding protein 4 abundances were shown to increase on both day 2 and day 6 at 2 different NPY concentrations. And lastly, lipoprotein lipase abundances were greater on day 6 with the NPY treatment versus the control treatment. NPY treatment appeared to increase expression of most pre-adipocyte and proliferation markers, while top 2A levels responded the most. Top 2A levels under NPY treatment were seen to be greater than the control treatment levels on all 3 testing days. Alternatively, the abundance of KLF1 did not respond to the NPY treatments at any dosage, and the KLF7 abundance levels decreased as a result of NPY treatment on day 4. As the graphs show, the greatest NPY expression was seen to occur on day 6 under the 100 nanomoles per liter treatment. As the NPY expression was related to the treatment, the NPY2R expression had no correlation to the treatments. These images, in addition to the oil red O stained images, display the increase in proliferating cells post-differentiation. These images are consistent with the gene expression analysis, strengthening the suggestion that 100 nanomoles per liter NPY treatment increased adipocyte proliferation. Figure A reveals that expression of NPY and NPY2R and other pre-adipocyte and proliferation markers all changed from 2 to 6 days post-induction. In particular, both GATA2 and KLF1 expression levels were highest right after induction to differentiation. Ki67 expression is similar to a bell curve. KLF7 expression was normal throughout, except on day 4 when the lowest expression was seen. Both top 2A and TP2X were seen to increase initially at day 2 and then decrease consistently until day 6. Lastly, Figure A displays the drastic decrease in NPY and NPY2R expression levels at days 4 and 6. In Figure B, the most striking finding was the drastic increase in steroregulatory element binding protein on day 4. Among the main conclusions reached regarding NPY systems and adipogenesis in avian species shown on the slide, this study also revealed that on average, higher dosages of NPY result in greater lipid accumulation and more gene expression changes for adipogenesis-associated factors. One aspect that can be taken from this study is the concept that the G3-PHD specific activity measurements were found to be able to be used as a gold standard indirect measure of adipogenesis due to the role that G3-PHD plays in relation to the differentiation of precursor cells into terminally differentiated adipocytes. Similarly, lipoprotein lipase was found to be an indicator for adipogenesis, and thus greater LPL levels supported NPY-promoted adipogenesis. Although this study was meticulously conducted, the time points between 4 and 12 hours used may have interrupted the maximum impact of NPY on adipogenesis by restricting its exertion on gene expression regulation in DNA synthesis. Furthermore, due to the relatively short half-life of NPY, the data had to be collected relatively quickly, and this may have unknown effects on the physiological mechanisms of NPY and consequently the values collected. A major consequence of this study is the application of this chicken model to study neurotransmitters' relationship to adipogenesis in birds, and concurrently from this, to examine the link between the central nervous system and adipogenesis in avian species, as well as across many different species. Reference: